Buonasera a tutti. Good evening, everybody. It's a real pleasure to be here again with this successful ISNAF New York Institute of Culture format of a webinar. Today, I want to thank you again once more, our acting director of the Institute of Culture, Paolo Barlera, for organizing and co-hosting through your platform this important webinar. And with, of course, ISNAF, and I do thank also President Chiara Manzini. And today we will focus on genetic disease, on very specific issues with great Professor Chiara Manzini, who is a very well-known and brilliant professor at Rutgers University here at the heart of New Jersey in our tri-state area. I can say that after the other webinar we had a few weeks ago on climate change, together with ISNAF and Institute of Culture, it's really a very important and eventful moment for scientific diplomacy. It couldn't come at a better moment because there's really a big momentum. Yesterday we had the big digital meeting of ISNAF with the ambassador of Italy to the US, Armando Barricchio, with a very important presence of our government, Minister Manfredi, and with you, dear President Zuffada, and with many scholars, many researchers, many physicians, many scientists that are really a big reason of pride for Italy in the US, a big reason also for being confident about the strength of our relation with the United States. I'm sure that also today, through this webinar, we'll have interesting moments, also through the brilliant wisdom of Maria Teresa Cometta, and I really thank her. Now, you really embody also this scientific diplomacy here in New York City. We're very grateful. And let me really confirm that this is not a peripheral topic or issue in diplomacy. It's more and more one of the core issues of our action. We are committed to a renewed and important scientific diplomacy. And as I have said also during the last webinar, I'm really willing, available as a Consul General here in New York to help the New York chapter, ISNAF chapter, to renew their association, their membership here, and we could organize all together a digital appointment the next January or February to relaunch our New York chapter. It's a strategic really field, and we want to cooperate whatever it takes as much as possible. Grazie davvero. I wish you an excellent seminar, and I give the floor to Cinzia Zuffada. Grazie.
Grazie, Console. Thank you so much for the opportunity for this second appointment here with the hashtag We Are ISNAF seminar series of 2020. This is the last for 2020, but it is not the last in our series. And so we hope that we will continue, and we will continue with a slightly different focus. And that is, we hope to catalyze interests at the regional level so that we might have the opportunity to share with the community that that work around and together with the consulate areas and Italian cultural institutes, and have a more targeted series to that community with its flavors in the presence of the scholars, the disciplines, the younger versus the more established. So we really hope to use this platform as a way of expanding our reach and tailor what we do to the audience we are talking to. So thank you for joining us today, and I look forward to a renewed connection with us in 2021. And with that, I'd like to leave this to the good hands of Maria Teresa Cometto, who is going to start the dialogue with Professor Chiara Manzini today. Thank you.
Thank you all. Thank you, Consul General, for your nice words. Thank you, Cinzia Zuffada, for having me as the moderator of your webinars. I'm so excited and honored to be the moderator of these fantastic conversations. And tonight, the title of the conversation is Adventures in Gene Hunting, Finding the Genetic Causes of Rare Brain Disorders, which is a fascinating topic. And we will see that actually we will not talk only about very rare diseases, but we will talk about what we learn about how the brain works. So all our brains, how they work. And we will talk also of not so rare diseases like autism, so which is something that affects more and more families, unfortunately. Dr. Chiara, what are the rare brain disorders at the center of your research?
So if I can show a couple of slides, I can show you, because it's easier if you actually see what those brains look like. And so we are particularly interested in intellectual disability, which is basically cognitive deficits. In some cases, we're also looking at autism. And so we are looking at brain disorders that may affect the structure of the brain, like this brain malformation that you see here. So this is a normal, would be a normal brain, and this child basically has an empty space inside the brain. And this is a very, very severe brain malformation called lissencephaly, or type 2 lissencephaly. And so you may end up with intellectual disability, and so with the not as, non-typical learning ability with a severe brain malformation, or you may have a brain that looks overall normal, but you still have cognitive deficits and learning deficits and autism. And the goal of my lab is really to try and find the answers for the families and the parents. And the questions that they have is this: how does this happen to my child? Will it happen again? And what can I do to help my child? And so just as an example, there are multiple types of brain malformations. And one of the things in thinking about rare brain disorders, or rare disorders in general, is that there is a particular mutation that is rare for any particular disorder, but there are more than 6,000 rare disorders that are known today that affect 10% of the US population. And so the disorder itself may be very rare and just affect a few people, but these genetic mutations are everywhere and may hit different genes in the genome, so that the burden on the healthcare system is actually pretty extensive. And so we primarily focus on these very severe malformations in the lissencephaly family, and on cognitive deficits that would have otherwise a normal brain.
Yeah, you focus also especially on genetic causes. What does that mean?
What that means is that we start from families, and so we identify families where we think there is a genetic condition, and then we sequence every single gene in the genome to try and find the one that is mutated and that is causing the disease. And so that's why I'm saying like gene hunting, and it's often an adventure, because it starts as like trying to find a needle in a haystack, and that is the answer to the first question: how did this happen?
Mm-hmm.
Right? Because families, in a lot of cases, these are very severe disorders. It's not just the brain, there will be other organs. They have a sick child, they have no idea what is going on. And often just being able to give a diagnosis a genetic diagnosis, which says this is the gene that is mutated in your family, is important. One, because the doctors may know how the disease is going to progress, and two, because they can find other families with the same mutation. And family groups in rare disorders are actually amazing organizations that coordinate the research and the clinical practice, and they're very, very important.
In practice, can you give us some examples of how you do this gene hunting? What tools do you use? What kind of research you do?
Yes. Let me show you another slide. Okay, let's stop. So we usually go to, we have to find the families. And so we have to find the physicians, and we have a very large network of collaborators. These are rare. So your average neurologist, for example, will see one of these kids in their entire career. So you have to find you have to find referral centers where these kids are going to be referred by their doctor that have experts that in these particular disorders. And so that's where we start collecting more and more and more cases. And so we collaborate with multiple countries all over the world to be able to identify these doctors that can find the kids. And once that you have the family, you take the DNA from everyone in the as many people in the family as you can get. You perform these kind of new, I mean, now they're 10 years old, but for us, they're still relatively new sequencing methods that allow you to sequence the entire genome. When I started in college, we were still, sequencing one genome, which took many, many years and millions of dollars. And now you can sequence a genome for $1,000 in like a few days.
Yeah, sorry to interrupt you, Chiara, but because you mentioned your college years, can we go back a little to your story, how you became interested in this field? Why did you decide to study this kind of disorders?
So I was always interested in how the brain develops. So one of the things that you can figure out from these disorders is actually what are the genes that are absolutely essential for our brain to develop, right? Because if they're mutated, if they're not functioning quite right, then the brain doesn't develop correctly. And so I was always interested in intellectual disability. And what's interesting is that when I was in Italy, I was actually, I was in the University of Pavia, which is one of the greatest centers for genetic research. I actually worked on infertility because the laboratory, I was an undergraduate, and the laboratory was like, this is what we need people to help with, so you're going to do this. And you're going to learn the trade, which is one of the things in science that often happens. You have to learn how to do things before you can do the things that you want. And so then from there, I really wanted to work on the brain. And I was very unsatisfied at the time that after you find a mutation, what do you do? How do you figure out what that particular change in the genome affects the brain? And so I came for my PhD to New York. I was at Columbia in a cell biology lab that was focusing on how the brain develops, just purely, not disease-based. It was entirely just looking at the development. And then from there, I went to Boston Children's and Harvard in a laboratory that was combining the genetics of these brain malformations with looking at development and really learning how to do that. And that's where I found some of the disease mutations that then my lab has been working on for the past couple of years.
Yes, and let's go on with talking about your gene hunting. So you said you find the family, you study the DNA of the family, and then
we study the DNA of the family, and then we go into different animal models.
I just really appreciated that.
And actually, I'll show you.
The insights were always there.
I think Cinzia is not muted.
Of course, right now, we can't tell whether you're in France or whether you're here. But the number of WebEx meetings that you will be on with JTLers will decrease.
Okay. Can we get, okay.
Okay.
Okay, so this is a good example of what we do. So, so we find a mutation, and then you have to go look into the brain. And so to do that, we don't do it in humans, but we actually recapitulate the genetic mutation in animal models. And we use both mouse and zebrafish. And so we use a combination of different models to do that. Did we get Cinzia muted? To do that. And then we use very advanced microscopy techniques where we can actually go and look at the cells and see what's happening in the cells when this mutation happens in the neurons, which are these like beautiful cells, when these mutations happens. And I think I have like a few pictures like this. These are neurons in the hippocampus, which is the part of your brain that controls learning. And these are stained with the very old techniques that Camillo Golgi, who was at Pavia, developed. And these are actually stained with like fluorescent methods and imaged with a laser to be able to look at the sites where the connections in the brain form, which are these little knobs that you see here in the cells. And so we do cell biology, and then what we also do behavior. So we figure out what is wrong at the cellular level, and then we ask the mice to perform different tests to figure out, are they learning okay? Do they have normal social function, especially if we want to look at autism? What are they doing? And the ultimate goal is that once that we identify that molecular change, then maybe we can find drugs that will restore the molecular changes and the behavioral changes, and those are usually called preclinical models. So you use an animal to try an experiment and figure out which drugs you could potentially use to then develop into humans.
Have you learned anything new that you didn't know about how the brain works during your research?
Well, what was very surprising, so we were developing a model of autism, and so up to a few years ago, all of these experiments that I was telling you about in mice were only done in males, because the female mice have an estrous cycle, which is like 4 or 5 days. And there was this overall perception in the field that because the females were cycling and the progesterone and the estrogen were at different levels, their behavior was unstable, which, you know, I'm not gonna go too much into that, but we could talk about it. And so all the experiments were being done in males. But then the National Institutes of Health realized that this was causing a huge amount of problems in clinical trials, because some compounds have different effects on women and men and males and females, and we were really actually not testing that. And so when we were doing all these experiments in male and female mice, we found a huge difference on how this particular mutation affected the males, which were severely affected, and they have cognitive deficits, social deficits, hyperactivity, and the females that only had some very mild cognitive deficits. And this was fascinating because autism and developmental brain disorders in general are mostly undiagnosed more frequently in boys than in girls. Mm-hmm. And it's very unclear why. There are a lot of labs that are trying to figure out, are boys more susceptible? Are girls protected? What is the difference? And so by studying this particular gene, we found a molecular difference that specifically affected males and didn't seem to affect females at all. And this mutation caused several changes in the male brain, but not in the female brain. And that was really exciting to me because it could mean that there are molecular differences in the male and female brain that could lead to this protection or susceptibility. And this is what we're studying right now.
So in a way, you confirm that our brain, I mean, women's brain is different from male.
So with the concept of the female brain and the male brain is wrong, right? The brain is the brain. But of course, I mean, hormones are different. There are several aspects of our development that are controlled differently in males and females. Evolutionarily, there are different roles, and this is particularly evident when you look at animals, for example, when you look at animal behavior. And so there are specific circuits and specific molecular differences that are set up to perform those roles. And it doesn't mean that one is better or worse, but in general, I mean, we know in humans, women have much better verbal memory and social function, and men may tend to have better spatial memory, but it's all on a spectrum. I mean, you can't say that one is, you know, absolutely not say that one is better than the other.
Okay, by the way, to all our friends who are following this conversation, please send your questions through the chat or through the question and answers app, because at the end of our first part of the conversation, we will collect your questions and Chiara will answer. Yeah, going back to what surprised you, what you found out. You have any theory about the reason why it happens, or you're just
you mean the reason why there is a difference, or the reason
no, it affects more the boys? You said already that there is this difference, but is there more about it?
So there are two theories that are being discussed right now in the field, and one is that females or women are genetically protected. So girls need more mutations to be affected than the boys. And why that happens, if it's epigenetic changes, and so changes that happen during development that change the way that your DNA functions, or other, we still don't know, and there's multiple groups, including mine, that is trying to figure that out. And then there's a second theory that elevated steroid hormones like testosterone and corticosterone during fetal development push the male brain to be more severely affected. And that is being explored also by several groups. Our findings that there might be molecular changes already present in the brain also raises the possibility that both of these theories are right, that the females genetically control the expression of these molecular changes, and that they can be pushed in the male to cause the disease. And so that's one of the things that we're trying to figure out. I can't tell you, I mean, right now, I don't think either theory is entirely wrong. I think it's a combination of the two.
Yeah, and what about the reason you include autism in rare diseases? Is it so rare? Yeah.
Well, so genetically, no, autism, I mean, right now, it's 1 in 58 kids. And actually what's interesting is that it's really, really high. It's much higher in New Jersey, which is also one of the reasons why I'm at the Child Health Institute of New Jersey, because New Jersey is one of the states in the US with the highest rate of autism. The problem with autism is that it's a constellation of multiple disorders. And so right now we know that there are more than 100 gene mutations that can cause autism. And some of them, the most common ones, will explain 1% of the cases. And so in fact, every single child with autism that has a specific genetic mutation has some kind of rare disorder that presents itself as autism. So we define autism as this like kitchen sink, basically, diagnosis. Like the kids have to have social deficits and restricted repetitive interests, which means that they can obsess on a particular toy, on a particular type of car, or they only eat a certain type of food, right? Which are the things that we usually hear in the news about kids with autism. But physicians that see these children will tell you that every child with autism is unique, has a very specific presentation and a very specific set of symptoms. And so in fact, autism is probably hundreds of rare diseases. And it's also possibly caused by environmental changes that we still don't understand.
Mm-hmm. Yes, earlier you mentioned that, of course, the goal would be to find therapies based
Right.
on the results of your research. At what point are you towards this goal? I mean, any kind of therapy has already been developed based on your kind of research?
So we are looking into some of these molecular changes that we find are being targeted for other types of autism in therapy development. And so we're really interested in continuing to develop them. So one of the issues actually with these disorders that, as I was telling you, for autism, there may be hundreds of different genes that cause a similar presentation. And so the question is, how do you develop a therapy when every single person has a different genetic mutation? And so you have to look for commonalities in changes in how the brain function or how the neurons in the brain are controlled, and try and see if you can combine as many cases as possible to develop therapies. And so we're trying to see what other researchers are doing with some of these molecules to be able to see if we can use it for the patients. In our case, our genetic cause of autism and intellectual disability, the gene that we study, which is called CC2D1A, is extremely rare. So we know of maybe like 30 people in the world that have that mutation. And so we have to be able to bring something to them. We basically have to be able to piggyback on other studies. I don't know if it makes sense. It's one of the complexities really in the therapy development for these disorders.
Mm-hmm. Yes, so you stress the fact that you need a lot of collaboration with other institutions. Do you collaborate with the Italian institutions on this kind of research?
I do. So we mostly collaborate on our brain malformations and neuromuscular disease because that's the part of our research where we're still actively doing a lot of genetics. And so we work very closely with the Istituto Besta in Milan. And as I was saying, we need these referral centers that are specialized in neurology where all the families go once that they receive a diagnosis. And so the Istituto Besta has very, very strong neuromuscular clinic. We've been working in the past with the Università Cattolica in Rome, which also has very, very strong genetics and neuromuscular research. And then sometimes we get other patients just because their doctor prescribes. So now you can prescribe this like sequencing approach where you sequence all the genes in the genome. And so when there's a child with a developmental disability that is unexplained, one of the things that they do, they just clinically order the test where they sequence all of their genes. And sometimes the gene that we study comes up and the doctors are like, we don't know what this is. We're not sure what's going on. And so the family will Google the gene and will find us. And then they will write us. So I've been in contact with some families in Italy through that.
Yes, very interesting. And well, is it fair to say that Italian scientists in neuroscience are, I mean, quite famous and important? I mean, that we have special excellence in this field. I just can remember that Nobel laureate Rita Levi-Montalcini won the Nobel because of her research on how the brain works, right? And there are many others quite famous. Do you think we have a special special gift about that, or there's a special school, tradition?
So Italy has always been very strong in anatomy and brain anatomy as a response. And so actually our first Nobel Prize for neuroscience was Camillo Golgi in 1906. Together with Ramon y Cajal. I mean, he made several other discoveries, but one of the first things that he was trying to understand was how the brain functions and how those beautiful cells with the staining that I showed you function. And Rita Levi-Montalcini has been instrumental in setting up like an amazing neuroscience school in Rome. You know, she had to leave during the war, so she was in St. Louis for a long time, and then came back and really established like very strong neuroscience research. Pavia traditionally has good neuroscience. Turin has, Università di Torino has amazing neuroscience, and Pisa. I mean, there are several places, and I'm definitely leaving somebody out that are doing very, very strong neuroscience in Italy.
In your lab, you have had Italian researchers.
Yes, I have had I have had multiple Italian researchers. I have hosted the PhD students. It's really nice that now in Italy, as part of your PhD, you have to do 6 months abroad, and it's part of your fellowship, and they pay for it. So it's really, I've hosted multiple PhD students from Italy for 6 months, which has been really nice. And actually one of them I met through the ISNAF annual meeting because our boss was participating and he sent me the student.
Great. So there is this exchange and collaboration going on. Very important.
Absolutely, yeah, absolutely. And I think it's one of the important aspects I think of ISNAF is really to build this bridge and continue this collaboration, and also provide the support to the Italian scientists that come to the US and really how to acclimate, how to figure out how things work. You know, when I came to New York, I was lucky because I had a friend that had graduated from Pavia right before and had come here. And her partner at the time was also somebody who worked in my lab, and they both moved to New York. And so I moved and I had friends immediately that helped me, but that doesn't always happen. Sometimes when you come from another country and you come to the US to do research, you're completely by yourself. And so the Italian community is often very important for finding your way and being able to get started with your life in a different place.
Yes. Do you have a message that you want to send to families with children with mental disabilities? What could you tell them?
Well, the first thing whenever I meet the families and I speak with the families is that I'm always inspired by their strength. The families of these kids are just amazing. All that I can say is that we're working for you. You know, we're doing our best, and we are really trying to understand exactly how these disorders are established, so that we can come up with solutions, and that we can help.
Yes. Well, before going on, I think we can check if there is any question from the audience. Please send your questions to the chat app or to question and answers, and we will try to answer your questions. In the meantime, waiting for any question, and going back to autism, there is a theory that many families in Italy have embraced about the connection between autism and vaccinations, or too many vaccinations. Is there any scientific hint that can be true?
Absolutely not. The original paper that supported the vaccines, the Wakefield paper, has actually been retracted, and several studies have been made to really explore this possibility. And usually what happens is that autism is diagnosed around 2 years of age. And not because it shows up at 2 years of age, is just that most of the instruments that the doctors will use to diagnose autism are only valid for a child that is beginning to speak and has certain abilities. And that usually coincides with a lot of the vaccinations that kids receive. And so a lot of people basically say, well, if you get the vaccine at 18 months or 20 months, then you're going to get autism. That is absolutely not true. We now know that autism begins in the womb. So it begins in the fetus, and it begins very early during development. And so vaccinations, childhood vaccinations, have absolutely no link to it.
Thank you, Chiara. Very clear your answer. We have our first questions. One is from Professor Nicola Di Cosmo. He asks, are the disorders that you study more common in some population groups than others?
Yes, in the sense that there are, so we tend to go in a lot of consanguineous populations. And I had a slide, but now I'm just not gonna. And so we work a lot with the Middle East. And one of the reasons why we work in the Middle East is that it was one of the places where you can still have a family with 8 or 10 children. When you're looking for a recessive disorder, like the disorders we study, where 1 out of 4 kids is going to be affected, if you have a family with 8 kids, you're going to have 2 possibly affected by the disease. And so we have been working a lot with countries where there are a lot of kids, but also where the parents tend to be first or second cousins. And that is part of their societal structure, where the marriages are often arranged. And so the arrangement happens in between families. And so in that case, it's actually even easier to find a genetic mutation when the parents are related. Usually the countries that have a high amount of consanguinity, like in the Arabic Peninsula or some areas in like North North Africa will have twice as much genetic mutations that in the normal, in a population like a European population where there is no consanguinity. There are also specific genetic mutations in populations that are enclosed and restricted. And so like the old order Amish that will only intermarry within the group. We have done some work also on the Ashkenazi Jewish population, where there are specific genetic mutations that basically just float around in that population that cause diseases that tend to be more common. But one point that I want to make, which was really interesting, like a few genetic mutations tend to be regional. So the average distance somebody traveled to find a wife up to like the 50s was 10 kilometers, because how far were you going to walk, you know? and so there are population specific mutations. And so we found one in southern Italy, for example, where there are mutations that originates in the past and then just kind of floats around and passes around in the population. So even if they're not first cousins, they may be 10th or 11th cousins, but genetic mutations tend to be regional. And then what happens now is that people start traveling all over the world and like marrying people that are very, very different from themselves, or having children with people very different from themselves. And so the genetic mutations move around.
Very, very interesting. Thank you, Chiara. Another question from Graziana Gatto. Hi, Chiara, great talk and interesting discussion. I had a question whether the gender-specific differences in autism also depends on the strain of the mouse, or if there is anything known about them being dependent on some generic background.
So that's a really interesting question, because in our case, we don't find that. So we actually looked at the strain of the mouse. And so for like the general audience, so laboratory mice are very, very genetically similar to one another. So the white mice and the black mice, and you know, the ones that you see in the news, were brought to the laboratory sometimes 80 years ago, and they all they're all mated with each other. They're all mated with each other, and so they're very genetically similar when you have one particular strain. And so those, then of course, every time you have a reproductive event, you generate mutations. And so each strain has specific mutations that kind of change the way they behave. So there are some laboratory mice that are very addictive. Like they love alcohol and cocaine, and they're more addictive than other mice. And there are some laboratory mice that are very calm compared to other ones. You know, kind of like if you think of your breeds of dogs and cats and like how different they are personality-wise, the laboratory mice tend to be like that. You get to know your strain. And so these mutations that lead to these behavioral changes could be accumulated next to other mutations that can change their susceptibility. In our case, we don't find it, but that raises the issue that it's very, very important when you do these experiments to know what strain of mouse you're looking at, because that genetic background could be really important for determining what you find.
Sorry, I didn't catch. What kind of mice have you chosen? Do you have different kinds that you study, or you have?
Yeah, we have. So we have inserted our own genetic mutation on several different strains. And so we will have it in a strain of white mice, in a strain of black mice, we have it in a strain of brown mice, exactly to try and get to some of those answers. Because when you breed your genetic mutation into a different strain, you can get different results.
Mm-hmm. And you didn't find any difference?
In our case, we haven't found any difference.
Mm-hmm. Yes.
Which means that it's a strong mutation.
I see. I'm trying to see if there are other questions from the audience, not at this moment. So what is your plan for the future? How long do you think will go on your research, or you think you will change your interest at some point after some results, or what?
No, because I mean, it's going to take us a very long time to figure out the brain. So what we're really hoping, so for the neuromuscular diseases, we are really going into like therapy testing and therapy development. And so we're making a lot of mutants, and then we're just going to try different therapeutic approaches and do a lot of drug screening. Neuromuscular disorders, differently from autism and intellectual disability, are having a huge amount of success with therapy development right now. And so we're very, very excited about trying different things related to gene therapy, but also to like drug treatments to try and improve brain and muscle function. For the autism and intellectual disability, we're just going to keep trying to figure out what are these differences in like molecular function between males and females. And I think there's probably like 50 years left if I live that long. If I want to be one of those professors that never leaves.
Great. Have you ever thought of going back to Italy, to Pavia, or to an Italian institution?
So I thought about going back to Europe, and so when I was looking for jobs, I really considered the possibility of going back to Europe. Italy right now is very complicated as far as like the research environment and what I have done. I mean, when I just moved to New York from Washington, DC. So when I was in DC, I was part of this group that was trying to provide advice to the government about how to improve grant funding, like have ideas on like how to develop better exchanges. And we were really trying to figure out a way to really improve the funding situation. I mean, right now, unfortunately, the funding for research in Italy is not very is not very good, and it's something that needs to change. And I was very excited actually when the minister spoke at the ISNAF event yesterday, and kind of like bringing this perspective that university and research have to become important for Italy again. I mean, the amount of money that we have in the US for doing research and that universities spend for maintaining the research infrastructure is much bigger. And I'm always in awe of the Italian researchers that do amazing work with whatever they have.
Exactly. I have a very light consumer kind of question to you. You earlier mentioned that at the beginning of your studies, it was very difficult and long and expensive to do the DNA genomic research, and now with $500 or even less, you can buy these kits and do your own DNA genome research. Is it worthwhile? Does it, I mean, for people? To
do the 23andMe?
Yes, stuff like that. By the way, there is also a very interesting startup here in America created by Italian researchers who does that. I don't know if you know. Yeah.
So I think so that is a slightly different thing, because one thing is like sequence all of your genome. What those companies do, they may sequence your genome, but also they look at your ancestry and they look at known changes in your DNA that may tell you where you're coming from, what kind of diseases you may be susceptible to. I think they're very attractive for Americans that tend to be a mix of different nationalities. And so because I know I'm Italian and it's just, I know where I come from, I wanted to find the German soldier that came through like 500 years ago that made me like blonde with like and 6 feet tall. And so I did the National Geographic test for myself, which was an older model that was looking at your ancestry and told you about how much Neanderthal you had, but also the migration patterns of your ancestors out of Africa, how long they had stayed in like Persia, and how they came to me. And I did found that I have 2% ancestral Scandinavian. So somebody must have come through a long time ago. They're fun. I think they're fun if you're trying to figure out your ancestry and where you come from. I think they're really interesting. I would be very cautious about the disease predictions. 23andMe got in trouble a few years ago because they were giving people a lot of information about possible diseases they would have without providing the appropriate counseling. Finding out that you're susceptible to some diseases can have very, very different, can cause very, very different reactions in different people. And you only know it when it happens to you. Like you can say, oh, I don't care. I definitely want to know if I'm going to have Alzheimer's. But what if it was for real? Right? And so you really have to work through, and I don't know if the tests we have right now are actually that accurate. So now there's a subset of disease predictions that they can come up with. But I would be cautious about those. But to look at ancestry and where you come from and which populations are in your genome, I think they're really, really interesting.
Okay. I have a question from the audience. I was reading yesterday about artificial intelligence models that try to solve the problem of, quote, protein folding, unquote, representation. Do you know anything about it?
No, I don't. I saw the paper that just came out, like looking at protein folding. And that would be a great question for one of the other members of the Scientific Council, Filippo Mancia, once that if he gives one of these talks, because he's a structural biologist, and that's exactly what he does. All that I know.
What is for people who don't know, like what is protein folding?
So protein folding. So the way it works is that, people usually think about your DNA as like a string. And when a protein is being made, the cell is reading the string and it's making a string of amino acids that just reading the DNA. These amino acids have to fold in some way to do what the protein needs to do. Because our proteins in our body are not just a string. And one good example that everybody should know is the spike protein on COVID-19, right? If you've all seen the pictures of the virus with those little bumps at the top. So those proteins have to fold accurately. And that is why we're doing an mRNA vaccine, so that the protein can be folded the way that it's supposed to be folded. And so there are some proteins that fold, I think all that I know, there are some proteins that fold in stereotypical ways, and those are easy to, or easier to detect with current methods. But if the protein has a disordered type of folding, it's a lot more complicated. So AI is being used for that. It's being used to try and understand the brain. It's definitely being used also for genomics. So we're all very interested in seeing what we can do.
Great. Another question, I think from a member of ISNAF. So what can ISNAF, the Italian scientist and scholar in North America Foundation, and your community in particular, what can you do to improve the situation of research in Italy? Given that yesterday the Minister of Research opened up to contributions from the scientist diaspora.
Right. Well, I mean, I think it's always so there are multiple ways, right? Definitely help with the globalization and like make sure that the students are exposed to all different kinds of like scientific research, and then can come back and like bring that information back, and then is just continue to do advocacy and bring the information into the government. It's hard because it's very different from what happens here in the US, right? So when I mean, I've been to Congress as a scientist to advocate for scientific research and met with like senators and congressmen, and you basically constantly talk and tell them what to do. In Italy, it's not quite as easy, but I was very excited when Elena Cattaneo was selected as a senator for life, and she has been doing a lot of work trying to bring science into the Senate and have scientists go speak to politicians. And so we just need to really make sure that they understand how important science is. I think right now, because of the pandemic, they are really beginning to understand. But I mean, the only answer that I have is to just keep going, and going at all levels, you know, make sure that the young people are trained and are able to go back and to bring new perspectives, but also to, you know, if you know anyone in government, to like make sure that they know what how important research is, and also develop more advocacy.
Yes, great. I think that we don't have any more questions from the audience, and we have talked a lot. We have covered a lot of topics. So interesting your research and what you are accomplishing, and especially what ISNAF is doing to foster research, innovation, and bringing together Italy and America in this effort to fight all diseases, rare and not rare. I don't know if President Zuffada wants to add anything, or Consul General. Otherwise, I say goodbye to everybody. Thank you for following us. And see you soon for other webinars.
Thank you, everybody. Thank you.
Buona serata a tutti.
Okay. Bye-bye.
Bye.